Inside every single living cell, there's a blueprint. It's a master set of instructions that dictates literally everything about it. And that blueprint, of course, is DNA. Now, before any cell, like our good friend the bacterium E. coli, can divide, it has to do something pretty incredible. It has to make a perfect, complete copy of its entire blueprint. And this is not a casual copy-paste job. Oh no, it has to be done with unbelievable speed and almost perfect accuracy. So today, we're going to peel back the layers and take a deep look at this amazing molecular copy machine in action.
So, here's our game plan for this deep dive. We're going to start by looking at the blueprint itself, the prokaryotic chromosome. Then, we'll figure out how the cell finds the exact starting line for this race. We'll watch as all the molecular machinery assembles and pries open the DNA, and then we'll get into the nitty-gritty of how the new strands are actually built. After that, we'll tackle the really clever solution to what I called the lagging strand puzzle. And finally, we'll cover the finishing touches, the cleanup, the proofreading, and how the whole process comes to a neat and tidy end. All right, first things first, before we can even begin to talk about copying DNA, we really need to understand what it is we're copying. In prokaryotes, like bacteria, the whole genetic setup is a little different from our own cells. For starters, there's no nucleus. Instead, all that precious DNA is packed into a specific region of the cytoplasm called the nucleoid.
Now, this bacterial chromosome has some really key features. First, it's usually just a single, giant, circular molecule of double-stranded DNA. For E. coli, that circle is made of about 4.6 million base pairs. Now think about that. How do you cram a molecule that long into a microscopic cell? Well, you use a process called supercoiling, which is basically twisting and folding the DNA up on itself until it fits. But the most fundamental principle we need to grasp before we go any further is the model that governs this entire process. Replication is semi-conservative. So what on earth does semi-conservative actually mean? It's a really beautiful and simple idea. When the DNA double helix gets copied, it first unwinds into two separate strands. Each of those original strands then acts as a template to build a brand new, complementary partner strand. So at the end of it all, you have two identical DNA molecules. And the cool part is, each of these new molecules is a hybrid, one old parental strand paired with one brand new daughter strand. This isn't just a theory, by the way. It was proven back in 1958 by the famous Maselson-Stahl experiment, which brilliantly used nitrogen isotopes to tell the old and new strands apart. Okay, so the cell has this massive 4.6 million base pair circular tract of DNA. This raises a pretty critical question. Where does it start? You can't just begin copying at any random spot. No, there's a very specific, designated starting block for this race, and all the cell's machinery knows exactly how to find it. And that starting block has a name. It's called the origin of replication, or for E. coli, OREC. That's OREC. This isn't just a random spot. It's a very specific DNA sequence, about 245 base pairs long, that basically acts like a big flashing "start here" sign. And within this OREC region, there are several repeating sequences called DNA boxes. Think of these as little docking stations perfectly shaped to be recognized by the proteins that are going to kick off this whole process. Initiation is this beautifully choreographed dance with several key steps. First, you have these initiator proteins called DNA. Fueled by ATP, they hunt down and bind to those DNA boxes at OREC. As more and more of them pile on, they form this protein complex that physically forces the DNA to wrap around it. This wrapping puts a ton of strain on the helix, and that stress causes it to pop open in a nearby section. And this section isn't random. It's a region rich in adenine and thymine bases. Why there? Because AT pairs are held together by only 2 hydrogen bonds, unlike GC pairs, which have 3, so they're much easier to pull apart. This initial opening is called the replication bubble. Next, another crucial protein, DNSC, shows up. DNSC is basically a specialized loader. Its entire job is to escort 2 donut-shaped helicase enzymes, also called DNAB, and load one onto each of the separated strands. Once the helicases are in position, DNSC leaves, and the helicases are now activated, sitting at the edge of the bubble, just ready to go to work unzipping the rest of the DNA. With the helicases loaded and activated, the initiation phase is officially over, and now the process shifts into high gear. Those 2 helicase rings start zooming away from the origin in opposite directions, unzipping the DNA double helix like a zipper as they go. This is what we call bidirectional replication. And each of those spots where the helicase is actively unwinding the DNA, that's a replication fork.
It gets its name from its characteristic Y shape. And since we have two helicases moving in opposite directions, we get two replication forks moving away from each other around that circular chromosome. They're destined to meet up on the other side. Now, unzipping DNA like this creates a couple of immediate, pretty serious physical problems that the cell has to solve on the fly. First, those single strands that have just been separated are perfectly complementary. Their natural tendency is to just snap right back together. Second, if you've ever untwisted a rope, you know that as you unwind one part, the part ahead of it gets wound up even tighter. This creates a ton of torsional stress, or what we call positive supercoiling. But don't worry, for each of these problems, the cell has a really elegant solution. So to solve problem number 1, the strands wanting to snap back together, the cell uses something called single-stranded binding proteins, or SSBs. The moment helicase separates the strands, these little proteins just swarm in and coat the exposed DNA. You can picture them as tiny little sentinels. They physically block the strands from getting back together, keeping them stable, separated, and totally accessible for the copying machinery that's coming right behind them. The second problem, that overwinding tension, is handled by an absolute master of an enzyme called DNA gyrase. It's a type of topoisomerase. To get a feel for what it does, imagine trying to unwind an old coiled telephone cord really fast. The part ahead of your hands gets tangled up into a knotted mess, right? Well, DNA gyrase prevents that from happening to the DNA. It zips along ahead of the replication fork and acts like a molecular swivel. It makes a quick, controlled double-stranded cut in the DNA, allows the helix to rotate and release all that built-up tension, and then perfectly seals the break. Without DNA gyrase, the whole replication process would literally grind to a halt in a tangled mess. Okay, so the track has been prepped, the strands are separated and stabilized, and the tension is being managed. Now it is finally time to actually build the new DNA. And this brings us to the star player of the whole show, the master builder enzyme itself, DNA polymerase. In bacteria, the main enzyme that does all the heavy lifting is called DNA polymerase III. Now, DNA polymerase III is an incredibly powerful and lightning-fast enzyme, but it has to play by two very strict, non-negotiable rules. These rules dictate the entire strategy for how replication happens. Rule number 1, it absolutely cannot start a new DNA strand from scratch. It needs a pre-existing starting point, specifically a free 3' hydroxyl group, to add the first new nucleotide onto. And rule number 2, it can only build the new strand in one direction, and one direction only, 5' to 3'. These two little constraints create some really fascinating challenges for the cell. So how does the cell get around that first problem, that DNA poly 3 can't start a new chain? It's pretty clever. It brings in another enzyme called primase. Primase is actually a type of RNA polymerase, and its job is to create a short, temporary little stretch of RNA about 10 nucleotides long right on the DNA template. This little piece of RNA is called a primer, and the very last nucleotide of that primer provides the free 3' hydroxyl group that DNA polymerase 3 desperately needs to get started. It's the perfect little welcome mat. Now for rule number 2, that synthesis only happens 5' to 3'. This one creates a huge logistical headache. Remember, the two strands of the DNA helix are anti-parallel. They run in opposite directions. This fact, combined with that strict 5 to 3 rule, means the cell has to use two completely different strategies to copy both strands at the same time. This is what gives rise to what we call the leading strand and the lagging strand. Let's start with the easy one, the leading strand. On this side, the template strand is oriented perfectly, running 3' to 5' into the replication fork. This means after a single RNA primer gets laid down at the very beginning, DNA polymerase III can just latch on and go. To make sure it stays attached and can copy thousands of bases without falling off, it gets locked onto the DNA by a ring-shaped protein called the sliding clamp. This clamp makes the polymerase incredibly efficient, allowing it to synthesize the new strand in one long, smooth, continuous piece, just following right behind the helicase. It's smooth sailing. But on the other side, we've got the lagging strand, and here we have a major problem. The template runs in the opposite direction. If DNA polymerase just followed the helicase, it would have to synthesize in a 3' to 5' direction, which we know it absolutely cannot do. So how in the world does the cell solve this puzzle? How does it build a new strand 5 to 3 while moving along a template that's pointed the wrong way? The solution is just brilliant. Instead of trying to make one long continuous strand, the cell synthesizes the lagging strand in a series of short, disconnected pieces. These pieces are called Okazaki fragments, named after their discoverer. In bacteria, they're about 1 to 2,000 nucleotides long. So, in essence, the cell makes this strand by doing a sort of backward stitch, making little pieces in the correct 5 to 3 direction, even though the overall process is moving the other way. So, how is all of this coordinated? Well, all the enzymes at the fork team up to form a single giant molecular machine called the replisome. And within this replisome, the lagging strand template actually forms a loop. This is really clever because it brings the two DNA polymerase III enzymes, one for the leading strand and one for the lagging, right next to each other. So as helicase unwinds more DNA, primase lays down a new RNA primer on that looped-out lagging strand. The lagging strand's polymerase then latches on and synthesizes a new Okazaki fragment until it bumps into the primer from the last fragment. At that point, it lets go. The loop is then reformed a little further up at the next new primer, and the whole cycle repeats itself. It's called the trombone model because the loop grows and shrinks, and it allows the entire complex to move in one direction down the DNA, even while one strand is being synthesized backwards in little pieces. Okay, at this point, the vast majority of the DNA has been copied. The leading strand looks great. It's one continuous molecule. But the lagging strand, well, it's a bit of a mess. It's all chopped up into pieces, with little bits of RNA primer stuck in between. So the final stage is all about bringing in the cleanup crew to polish the work, check for any mistakes, and bring the whole process to an official end. And this cleanup crew has two star players. First up is DNA polymerase I. This enzyme has a special trick up its sleeve that Pol III doesn't have, a 5' to 3' exonuclease activity. This lets it act like a little Pac-Man. It moves along the strand, chewing up the RNA primers in front of it. And as it removes each RNA nucleotide, it uses its other hand, its polymerase activity, to fill the gap with the right DNA nucleotide. It's a remover and a replacer all in one. But after Pol I is done, there's still one tiny little gap left, a break in the sugar phosphate backbone called a nick. And that's where DNA ligase comes in. It's the molecular glue. It uses a bit of energy from ATP to form that final chemical bond, sealing the nick, and finally joining all the Okazaki fragments into one beautiful, continuous DNA strand. Now, something incredible has been happening this whole time that we haven't talked about yet: quality control. Both DNA polymerase III and I have a built-in proofreading function. As they're adding new bases, they're constantly double-checking their work. If one of them accidentally puts in the wrong base, the shape of the helix gets distorted, and the polymerase just stops in its tracks. It then uses its other exonuclease activity, this one runs 3' to 5', to go backwards one step, snip out the wrong base, and have another go. It is literally like having a delete key built right into the enzyme. And this system is just unbelievably effective. Without proofreading, the polymerase would make a mistake about once every 100,000 bases. Not bad, but not great. With this real-time proofreading, that error rate drops to about 1 in 10 million. But it doesn't even stop there. Other repair systems come along after replication is done to scan for any mistakes that might have slipped through. And when all is said and done, the final error rate is a truly mind-boggling 1 in a billion. That kind of accuracy is absolutely essential for keeping the genetic code stable from one generation to the next. So what happens when those two replication forks, which have been traveling around the circle, finally meet? Well, it's not a chaotic crash. It's a highly controlled stop. On the opposite side of the chromosome from the origin, there are specific stop signs called TER sequences. A protein called TUS binds to these TER sites. And this TUS-TER complex is ingenious. It acts like a one-way gate. It will let a replication fork pass through if it's coming from one direction, but it will physically block the helicase if it approaches from the other. This creates a fork trap, ensuring the two forks meet right where they're supposed to and don't just keep going around and around. Once they're both halted, the cleanup crew does one last pass. And finally, the two brand new circular chromosomes, which are now linked together like rings in a magic trick, are separated by another topoisomerase into two perfect, distinct daughter molecules. And so there you have it, a process of just incredible complexity, coordination, and precision. From one single starting point, this amazing team of molecular machines, the replisome, works together to copy an entire 4.6 million base paired genome. They do it at a rate of thousands of bases every second, with an error rate of 1 in a billion, and they get the whole job done in under an hour. It really is a masterpiece of molecular engineering, and it's happening countless times a day inside every single dividing bacterium on our planet.